Hi Sanchita. So we are doing this for
Hello.
Pulse, and congratulations for your recent publication. So we have this called as Potion of Science, and the research spotlight is on you for this month.
Okay.
So can you start with introducing yourself?
Yeah, so I have done PhD with Dr. Bhupesh Taneja, and I have done on a dermatophyte, Trichophyton rubrum, and try to study its pH-dependent regulatory pathway. Uh, in the study, I tried to evaluate and understand a transcription factor PacC, which plays a pH regulatory transcription factor in this dermatophyte or the fungal pathogen, that is Trichophyton rubrum. So Trichophyton rubrum is a dermatophyte. It causes skin diseases on human beings.
Regarding this paper, so can you just kind of summarize the findings that were there in this particular article?
We used a CRISPR-Cas9 based NHEJ based technique to kind of create a knockout of this pH regulatory PacC transcription factor to understand how it affects that organism. So we have used a dual-plasmid-based CRISPR-Cas9 technique uh, by which we created knockout of this particular pathogen and tried to study or characterize its functional relevance, how it affects the pH regulation and other pathways.
Right. So I think the other day when you were just talking, you said like it was kind of a difficult journey for you. So do you want to kind of share something related to the paper or otherwise, like your PhD journey?
Related to the paper, I would like to narrow it down because my PhD has been a very long tenure. Usually unusually wrong or usually wrong long. It took more than 7 years maybe. And with respect to this paper, this was close to my heart because I started in a structural biology lab under Dr. Bhupesh Taneja, and in my say last after say fifth year or so, we realized that we could not try getting the crystals for that particular protein. So the paper is about CRISPR-Cas9 for creating knockout of a particular gene, and this was one of my objectives and not the main paper. But the PhD turned out in such a way that eventually this became the core, and then we shifted to transcriptomics and other studies and biophysical studies as a sub-objective. So I redesigned in trying to redesign my PhD. This kind of became the main focus of my study and not a side one. So this paper meant a lot to me, the study actually. And I belong to a structural biology lab without any prior expertise in the field of CRISPR-Cas9, which usually are say the strength of other labs. It was therefore tough to kind of standardize, understand, optimize the system in the fungus for the first time. And I did it with the help of under guidance of Dr. Shivprakash Ramalingam, without whom I don't think this paper would have been possible. So along with my guide, Dr. Shiva helped me design and execute and plan the experiments and then publish it.
Right. I think that's one thing which most people who work with protein crystallization face. I think like what you face in the final year, suddenly, I mean, you have to change your project. So yes, I think it's a difficult journey, and congratulations for such a commendable work.
Thanks.
And you could start a new technique in the lab and then actually take it forward until your paper.
So yeah, so I was the first student to start this, and now like I feel good that many of my juniors have been carrying this technique and trying to understand other genes and pathways in the same system. So it it feels good to know this.
Right. So you've already shared like how you felt good after the paper. So what about during this journey? Do you want to share like any memorable moments? that you are going to stay with you?
Not particularly memorable. See, this was because this was not our forte or strength. Everything that was new and every result. So whenever we got the results, it's not that we were used to getting those or we interpreted it very well. So there were
Hmm.
hurdles, and even in trying to understand, we grew as a team. So me, my PI, we learned new things.
Hmm.
We discussed it with Dr. Shiva to understand it better, because Dr. Shiva specializes in mammalian cells and CRISPR, not fungus. So three
Right.
of us worked together, discussed together to understand it. And many of my colleagues from IGIB, those who belong to the CRISPR lab in particular, say Debo's lab or Shiva's lab. So these
Hmm.
people also play a major role for discussions and building up a project, helping me with many difficulties which I faced. So every step was important, and discovering that
Hmm.
okay, this is going right. Here it needs to be worked out, and then how to overcome that issues.
Would you want to share like what you used to do during your PhD life, like outside the lab?
Usual outside the lab, I'm into music, and I mean, I like singing, reading. So then we have
Right.
times, good times with our lab and friends
Hmm.
and colleagues. We party, we celebrate. So those are the occasions, but mostly it is has majorly to do with work.
So, coming back to work again then. So, like usually it's said that how, it's always the reviewer number two who's going to
Mhm,
kind of create some hurdle for passing through of your paper. So,
mhm.
did you face any such, funny or any such comments which could not be addressed or which could be addressed and actually made your paper better?
So this paper, which has been published in the Journal of Fungi, it had been sent to another publication, another journal, but it took almost. So this was submitted during the Corona phase two, but it took almost one complete year, more than a year and a half. And then
Hmm.
at the end, that particular journal, I'm not naming the journal, it did not reply back as to whether it had
Oh.
any progress in the revision. So we waited for quite a long time, and then we ended up taking that paper back, I mean, and sending it to a new journal, that is this Journal of Fungi. So this was pretty fast for us. It worked very well. The reviewer number two had issues, but those were very technical, straightforward, and pertaining to the
Right.
questions which he found or he did not understand. So those were answerable or
Hmm.
addressable. And luckily, this was very straightforward for us. So it took almost a few months, maybe one or two, hardly, for it
Ah,
to
that's fast.
get submitted and then get accepted altogether. So the reviewer was sweet, whosoever it was. He was really sweet.
So you, I think, completed your PhD this year only
Yeah,
in
I did it
October.
in October. Yes.
Yes. So what are your plans now?
So I have already applied for a postdoctoral
Okay.
position, and I've got in one of the universities in USA. So
Okay.
I'll be joining.
Congratulations.
Yeah, I'll be joining there in probably May and say
Okay.
2023.
Nice. So it's going to be like similar work that you've been doing here or
It is
something new?
so like I said, my this first publication wherein I had first author. So
Hmm.
this is my first publication wherein I am a first author. Uh, this was a CRISPR based paper. My
Right.
future work is will be dealing with CRISPR Cas9, but
Hmm.
it will be on a mammalian sister. So it will be on
Okay.
cardiomyocytes.
Thank you so much for talking
Thank
to
you.
us, and I
Thanks
wish
a
you
lot.
all the best for your future endeavors.
Yeah, thanks a lot.